start, but like I said, I'm going to let everyone know that the recording is attendees are starting to join us now. We have about 5 that are coming in, 6. Good morning, everyone. Happy Saturday. We'll wait a few minutes and let everybody join us. If you've been on a Zoom call before, this is a Zoom webinar, so you will only see the panelists, myself, and the panelists from LogicBio, and your attendance is for viewing only and chat. I'll wait for a few more people to get on and I'll explain how the chat feature works. My name is Kathy Stagney. If you haven't already met me or know me, I am the Executive Director of the Organic Acidemia Association, and I welcome everyone. I'll wait a few more minutes. Hopefully everyone's not having any difficulties dialing in, but Zoom can be kind of tricky sometimes, depending on the connection. But for the most part, I think it's been a pretty good tool to use during this pandemic, where a lot of people need to have connections, and this is a good way to connect with folks. It's probably the only way I see people these days. About 16. I think we had about 23 or so signed up total. So there may be some that will pop on a little later. I know Saturday mornings can be a little crazy depending on where you're calling from. If you're on the West Coast, still could be a little early for you. I'm in Minnesota. Again, my name's Kathy Stagney. I'm the Executive Director of the Organic Acidemia Association. And welcome everyone this morning to the LogicBio webinar. We have some panelists from LogicBio that will discuss their upcoming clinical trial, Sunrise trial they call it. We have about 17 on now, so I think we'll go ahead and try to get started. This is going to be recorded, so if you want to come back, I will post it on our Facebook group and on our YouTube channel. Or OAA has a YouTube account, and so it'll probably be there for future viewing. And if families, there will probably be some that would not be able to attend today, or you want to go back and revisit. I would just give you a brief introduction of LogicBio. I heard about LogicBio back in 2017. I got an email from Kelly Blackburn, who at the time was their patient advocacy person, that wanted to let me know that they were working on clinical trial for gene therapy for MMA. And I had the privilege of going to Boston and meeting the team. Me and my family went last November. Well, actually it was November 2019, my last airline trip prior to the pandemic starting. So we went and visited. I gave a presentation on the Organic Acidemia Association, and just to kind of give them an idea of what we do here. And I've put multiple articles and updates on their work on the clinical trial in the newsletter and on Facebook. And they're going to share, updates on where they're at. They're hoping to get this all started this year, so it's very exciting. I'm going to pass it over to Jamie Ring, who is their patient advocacy coordinator, that's going to share and introduce you to their team. And like I said, we're going to have a polling question first that I'm going to share with everyone. It's about 4 questions. If you feel comfortable answering them, they're pretty just straightforward to kind of give everyone an idea who's on the call today. And I'll give you a couple minutes and have everybody answer these questions. that I'm going to show. Hopefully everybody will see those. And we have a chat feature down on the bottom of the screen, and feel free to, if you want to share where you're calling in from. And the Q&A feature on the bottom of the screen is where you can share if you have a question regarding what is being presented today. Feel free to put your Q&As in there, and they will answer those at the end of the presentation today. So everybody can see the polls. If you don't feel comfortable answering, that's fine. I will show the results here in a few minutes. I'll give you a couple minutes here to answer these questions.
Right. I sent the email to register. I'm waiting for a response.
Well,
with the link. Who does it go to?
Me?
Well, other people might be trying to get in.
No, I've already tried.
Give me the passcode. Okay.
Okay. I got 73% has voted. So I'll give you just 2 more seconds here. If you feel like answering, if you don't feel like answering, that's fine too. But I'll end the polling. I'll share the results and say it seems like most people have heard of, 50% have heard of LogicBio. Gene therapy, they pretty much know a little bit about it, still learning. That's pretty typical. Interested in participating in the clinical trials, perhaps. And when I think about treatment for my MMA, a lot of them are hoping for something new and help better manage the disease. But okay for now. Okay. So that kind of gives a little idea of what's going on with that. Okay. So I will hand it over to Jamie, who will do the introductions, and we'll come back in about an hour. We expect this to last maybe an hour with Q&A. And like I said, if you have any questions, the Q&A chat is on the bottom of your screen. Feel free to share that with the panelists or with everyone. If you have any questions, they're going to answer those at the end. And if you want to put in the chat where you're calling from and share who you are, you're open to do that as well. So I will hand it over to Jamie.
Great. Well, good morning, everybody. And we want to thank you for taking time out of your day to join us and to learn a little bit more about LogicBio and our work in MMA. We especially want to thank Kathy and the OAA for giving us this opportunity to spend some time with you and provide an overview of our research and, as Kathy indicated, our upcoming Sunrise clinical trial. We have a number of slides that we're going to review with you today, but really want to also hear from you and help address any questions that you might have. So as Kathy indicated, please feel free as we go along to enter your questions in the Q&A, and we'll do our best to address those. As Kathy said, my name is Jamie Ring, and I work with LogicBio in the patient advocacy function. And patient advocacy, for those who may not know, our role and our goal is to really serve as an ambassador for LogicBio out to the patient communities that we are conducting research for. And importantly, also serve as a liaison for the community back into our organization. It's really important for us as a drug development company to better understand your perspectives as family members who are affected with diseases that we're researching, better address some of the unmet needs that you have. And we believe that our best ability to hopefully bring potential treatments to rare disease communities comes from partnering with the patient communities that we're looking to serve. So like I said, we're really excited to have this opportunity, and I'd like to introduce or invite our other colleagues to introduce themselves before we get going. So Marie, could I ask you to introduce yourself?
Sure. Hi, everyone. I'm Marie Payton, and I work in the clinical operations group at LogicBio. And that means I work with all the different hospitals and doctors to get the clinical trial up and running and help to support them in anything they need to help treat the patients on the clinical trial, answering questions, doing any training, making sure they have supplies, and then ultimately working with regulators such as the FDA to get the drug approved at the end of all the research that will need to be done. Joined LogicBio about 2 years ago, and I joined them because it's just such a compelling need that this patient community needs. So really excited to get the trial running and to really bring a potential new therapy to patients that need it.
Thanks, Marie. And then Dr. Daniel Gruskin, he's going to be providing most of the presentation today, but if you want to go ahead and introduce yourself.
Sure, Jamie, thank you. Hi, everybody. Thank you so much. I'm really grateful to you for giving up some of your precious time on a Saturday morning or Saturday afternoon, depending on where you are, to hear about LogicBio, to hear about the work that we're doing to hopefully develop a new treatment for MMA. As you've heard from Jamie and Marie, we're really excited about this potential and the chance today to share some information about it with you. As you've heard, my name is Daniel Gruskin. My position at LogicBio is Head of Clinical Development. Like Marie, this is the group whose job it is to design the clinical trials and conduct those clinical trials that determine whether a medicine works or not. I've been with LogicBio for about 6 months now. My background is in medicine. I'm a doctor. I don't practice anymore, but when I did, I was a clinical geneticist. Probably most or all folks on the call today have a geneticist as part or the leader of their care team. I'm from Atlanta. I practiced in my hometown at Emory, where we had a fairly busy genetics clinic, and I helped take care of patients with metabolic disorders like MMA. And so I'm really happy to be talking to you all today to share the work that we're doing at LogicBio. But as Jamie mentioned, I would also be thrilled, I think we all would be thrilled to hear from you afterwards, to learn from you, because it's really critical for any company, but particularly those in the rare disease space, to really get information from the patients and the families that they're trying to help. So we know that we're trying to address the things that really matter and that are important to you. So thanks again for your time today. Really excited to talk to you.
Great. So I think we're going to go ahead and pull up our presentation and get going. And thanks to those of you who have introduced yourself. I see that we have folks really from across the globe and across the country. So thanks again.
Is the screen showing?
It is.
Jamie, did you want me to speak to this?
Yeah, if you want to go ahead, you can go ahead and take this.
Okay, so first what we wanted to do was just introduce you to LogicBio. I saw from the survey that some of you have heard of us, but maybe don't know too much about us. Others have never heard of the company at all, so it's a good opportunity just to tell us a bit about who we are. We are headquartered in a town outside of Boston called Lexington. We were founded in 2016, and we are, as the slide says, dedicated to providing genetic therapies that are given a single time with results that last. And we'll talk much more about that bullet over the course of the presentation, what exactly that means and how we're trying to do that. We are focused right now on rare diseases, and particularly those rare diseases that affect children. We're really passionate about addressing an area where we feel maybe has been neglected, rare diseases, and particularly young people with rare diseases. And you all know our first program, our lead program, is in methylmalonic acidemia for several reasons. We think this is an important disease to try to address. And as we go through the presentation, I think you'll see some reasons why we think that. We're relatively small, about 40 people. Most of them are scientists working in the lab, but we are growing and increasing our numbers almost on a daily or weekly basis. Many of the people within the company, and certainly the leaders of the company, have rare disease experience and rare disease passion, which is more important. Really care about helping people who have rare diseases that may other large pharmaceutical companies may not be spending much time thinking about. You see pictures at the bottom. 3 of the 4 of us are on the call today. Fred Chareau is our CEO. He wasn't able to join today. Fred has many years of experience in rare diseases at GeneRide, a company you may have heard of, really a pioneer in rare diseases, and other companies. And then you've met the rest of us today. So that is the team. Just quickly, the agenda for today. The ultimate goal really is for us to tell you what we are working on for MMA families like yourselves. But really, in order to do that, we thought it'd be important that we were all speaking the same language, that you all had a good understanding of the basics of genetics and gene therapy. So, we're not using terminology and talking about things that are confusing and maybe difficult to understand. So I will be spending a lot of time, at least at the beginning of the presentation, giving some background information on genetics, so you'll be able to interpret what the heck we're talking about when we talk about the trial and the technology itself. I'm guessing that some of the material will probably be a review for some of you guys, especially if you have a science or a medical background. But if you'll bear with me, I think it's important that we get everybody on the same page before we dig into the details. And so we'll go through genetics, through gene therapy, and then our specific technology, which is called GeneRide. And then we'll talk about the basics of clinical trials themselves, and then about our clinical trial before heading into a Q&A. So the first thing I think we need to do is just, again, make sure we're speaking the same language, the beautiful language of genetics. And here's some terminology that we'll be talking about today, starting with cells. These are the building blocks of your body. Our bodies have trillions and trillions, about 30 trillion cells in the human body. They come in lots of different types depending on where they are in the body. There's brain cells, liver cells, blood cells, etc., and they each do different things. Proteins, I know that you know this term, but probably most people think of it in terms of diet. But inside the body and inside the cell, a protein is a molecule that does most of the work of the cell. It's made up of a string of smaller molecules called amino acids, and some of these proteins are called enzymes. These are specific proteins that help do chemical reactions. DNA is the genetic material inside the cell. You can think of it as a code. It's a set of instructions that tell each cell which proteins to make, depending on the kind of cell it is. So all the cells have the same DNA, but different parts of the DNA is active depending on the cell type it is, and that tells the cell which kind of proteins to make. And it's the genes, which is a specific part of the DNA that does that, that tells the cell what specific protein to make, and different genes are active in different cells. Genes are collected in structures called chromosomes. These are inside of every cell and are just the structures that contain the DNA. And there's another molecule called RNA that you've probably heard of if you've been paying attention to COVID vaccinations, because some of those are made up of RNA. And this is part of the process between DNA and protein. And so now that we have those words defined, on this slide, I'm showing the path from the cells to the proteins that they produced. And as I mentioned, there's lots and lots of these cells in our bodies. And inside each of those cells are these structures. You see they kind of look like X's. Let me get here we are, that are called chromosomes. This is what they actually look like under a microscope. In this, they're not nicely arranged like this. So in this picture, they've been cut out and rearranged by height. And as it turns out, they come in pairs. There's 46 of them in every one of our cells, 23 pairs. And when you think about it, it makes sense that they come in pairs, because we inherit one copy of each chromosome from our mother, one copy from each chromosome from our father. And so 23 from each parent form this set of 46 chromosomes. Scientists knew about these chromosomes a long time ago. In the late 1800s, they were discovered, but didn't know what they were or what they did until a few decades later, when they realized there was a link between these chromosomes and inherited traits like eye color and gender, etc. And it turns out that these chromosomes are actually made up of this long structure called DNA, very long strings of DNA. And a good way to think of DNA as a string of letters. It's 4 letters, in fact, T's and C's and A's and G's. And sort of like how a sentence in English, if you mush all the words together, it's just a string of letters. That's what DNA is, a string of letters that forms the code of DNA. And the cell knows how to interpret this T, A, C, C, A, C, A, etc. into instructions on what to do. And that what to do is the proteins that they're supposed to make. Now there's a step in between DNA and protein. This is the RNA. The DNA is converted to RNA. I only include it here because it shows that the code is actually made up of 3-letter triplets, sort of like the words in the sentence. And each triplet, each 3 letters, codes for a specific amino acid, which is what, as I mentioned, proteins are made of. So this AUG tells the cells that it needs to start with this amino acid called methionine. This GUG tells the cell, okay, next you need to add a valine, and then et cetera, along down the line, and eventually you have a full protein, a string of amino acids based on the DNA. And I use this roller coaster as a metaphor for a protein. I think I like it because a protein is a 3D structure. It's not just a line. It has folds and curves and bends, and the shape of that protein is dependent upon exactly the order of these amino acids, and how they interact and how the amino acids are shaped determine the eventual shape of the protein. So that's how it works when everything is going well, but what happens if something goes wrong? So mutation can be kind of a scary word, but it really just means a change in the code of the DNA. And for example, if a letter in the code is replaced by a different letter, the word, so to speak, is misspelled. And when that code word is misspelled, the cell then is told to do the wrong thing, basically adding the wrong amino acid to the protein. And so here you can see part of a gene, the letters forming the code. These letters here, AAC corresponds to asparagine, TGC codes for cysteine, etc. And when everything goes well, the amino acids are in the right order, and you get this nice working roller coaster. Here I put a circle here to sort of denote these three amino acids form the top of that hill, a rise and then a bend and then a fall. But if there's a mutation, this T is a C, then you have a new triplet code here. A TGC is now a CGC, and CGC is the code for arginine, not cysteine. The cell doesn't know that there's been a mistake. It just does its thing and creates this new amino acid sequence, this different protein. And potentially this amino acid has a different shape than the cysteine, and you end up with a straight line instead of a rise and a bend. And clearly now that roller coaster is not going to work. And the metaphor is it creates a protein that is not, doesn't do what it's supposed to do. By the way, I just want to remind everybody that there's the Q&A box. So if there's any questions about anything I say, please feel free to add a question and we'll try to address it at the end. So now let's talk specifically about MMA. I'm sure that you've seen a version of this pathway before. I just wanted to talk a bit about how the genetics relates to the cause of MMA. So our body breaks down proteins, whether we eat a piece of chicken or a fish, or if we're not feeling well, if we have a viral illness or are fasting, then our body needs energy and amino acids in a different way, and it breaks down the proteins in our muscle of the body. These amino acids are then broken down further, going through a few different steps to methylmalonyl CoA. This is just another molecule along the pathway. And then the mutase enzyme is what breaks down methylmalonyl CoA, and eventually the body gets energy from that. By the way, mutase, for the enzyme to work, it needs vitamin B12. This is why many patients with MMA are on B12 supplementation. But if there's a mutation, a change in this gene, the mutase gene is denoted with an abbreviation MMUT. If there's a mutation in that gene and you end up with that changed roller coaster and that protein doesn't do what it's supposed to do, then there's a block in this pathway. And when you take in protein from food, or you're sick and you're breaking down your muscles for energy and protein and amino acids, it's the pathway stops here. Methylmalonyl CoA gets created, but can't go further down the pathway, and it builds up. When there's enough of it, it gets converted into another molecule called methylmalonic acid. This, unfortunately, is toxic to the human body and causes the disease, methylmalonic acidemia disease. So this is a slide I show to doctors and others when I'm teaching them about MMA. The people on this call are already experts in the disease, so I won't spend too much time here. You're well aware it's a rare disease. About 1 in 50,000 babies are born with it in the US. Luckily, it is on the newborn screening panel in every state in the country. And it's estimated that about 1,000 or 1,500 people with MMA are living in the United States, and about the same in Europe. But even though it's rare, we feel that there's still a tremendous need for better treatments. And we saw in the survey at the beginning of the presentation that folks on the call also are hungry for new and better treatments, and that's what we're working towards and are talking to you about today. You know better than I about the difficulty in maintaining the restricted diet of low protein and the lack of available therapies to really address the underlying metabolic cause, that the enzyme defect in MMA. And because in most patients, the diet doesn't completely eliminate the symptoms of MMA, transplants are being increasingly performed in children with MMA, and they're being performed at younger ages than they used to be. Transplants do appear to improve things, but of course it's a major surgery. It has its own set of issues. So research is taking place at LogicBio and at other companies, which is great. I'm always happy when more companies are getting into rare diseases, and academic institutions as well, like NIH, are trying to find a new treatment, including gene therapy. And so now let's talk a bit about gene therapy itself. So gene therapy, I'm sure, is something you've heard of. It might sound a bit like science fiction, but it's actually been around for quite some time. Chromosomes, as I mentioned, were discovered in the late 1800s. And it was in the 50s when scientists figured out what exactly DNA was and what it did. And it only took a few years later when a virus called AAV, adeno-associated virus, a virus that's not harmful to humans, was discovered, and scientists realized that it could be used to deliver genes that were created in a lab, working genes, could be used to deliver those into cells. Basically, before that, gene therapy was an idea. People thought it, was a potential thing that could work, but the discovery of AAV really made scientists realize that it could actually be possible. It did take a while after that to work out the details, but in 1990, the first gene therapy trials were initiated. And in 2012, gene therapy was actually approved in Europe. This was the first gene therapy approved anywhere. It took a few years later for FDA to approve a gene therapy, and as of today, the number, frankly, is kind of shocking, but over 2,600 gene therapy clinical trials are going on or have been completed. A second gene therapy has been FDA approved. There's a lot of work and excitement going on in gene therapy, and it holds a lot of promise for many diseases, but in particular rare diseases, which many of which are caused by a single gene. So what exactly is gene therapy? So now we've gone through what a mutation is, what it does to a gene to make it not work. And the idea with gene therapy is to replace that gene that doesn't work, or that's creating a protein that doesn't work, with a good one, with one that does produce the right protein, or to fix that gene to make it work. There are several different types of gene therapy. I just wanted to show two of the main approaches people are working on today. This is called gene replacement. This is probably what most people think about when they hear the term gene therapy, where a new working copy of the gene is delivered by this AAV virus. What's normally inside the virus is scooped out and replaced with the gene that we're interested in. This virus goes to the cell and delivers the working copy of the gene, which is then goes through that protein factory process. One thing to note here is that it doesn't get incorporated into any of the chromosomes that are already in the cell. It is sort of floating outside of those chromosomes. This is an important detail to understand, and I'll show you why that is in a minute. Another type of gene therapy that has a lot of excitement around it these days is gene editing. This is where the DNA on one of these chromosomes that's already in the cell is actually changed by the treatment. This typically requires what's called a guide molecule that matches up with the DNA that's already there. So it brings the treatment to the right place on the chromosome. And then a special enzyme that cuts the DNA, that cuts out a piece of DNA, and then either reconnects the cut ends or replaces it with new DNA, but actually changes the DNA that's on the chromosome that's already there. So this works a little differently than traditional gene replacement. It's incorporated into the natural chromosome needs this cutting enzyme, also called a nuclease, in order to do that. So why am I making that distinction? And it really, among other things, has to do with how cells replicate. This process is happening in all of us as cells need to replace themselves, but especially in kids as they grow and they need the tissue to grow, and they do that by having these cells multiply. They divide and make two cells out of one. But each of these cells needs a full copy of the DNA, the instruction manual, in order to do what it needs to do, to make those proteins, to make it a liver cell or a brain cell or whatever kind of cell. And this is how that happens. I'm only showing one pair of chromosomes here for simplicity, but you can imagine there's 46 of these in a cell. And first, these cells, these chromosomes copy themselves. They make a duplicate like a copy machine, and you end up with chromosomes that look like this. This is why under that microscope it looked like an X, because it had already been copied. And then these cells sort of pull the chromosomes to each side, pinch together, and you end up with two daughter cells with exactly the same DNA makeup as the original cells. In traditional gene therapy, however, remember there's this genetic material that we delivered, that was delivered and stays outside of those chromosomes. And so when that cell replicates, when it's time to grow by dividing, only one of those cells gets that DNA. It's not copied like the rest of the chromosomes. This happens, you can imagine, multiple times. And so after the first round, there's one of the two cells has that genetic material. After another round, one of four, and so on. And so what has been seen in some of the gene therapy trials that uses this technology is that there's a dilution of the effect, meaning it starts off having a certain effect, working well, but then as time progresses, because these cells are multiplying and the number of cells with that working gene make up a smaller percentage of the cells, the effect decreases. This is something that has been seen in other studies. The FDA is very interested in this effect. Nobody wants a treatment effect to reduce over time. With gene editing, however, this really isn't an issue. You know why now? It's because the DNA on the chromosome is actually being changed. So that change is carried on with each division in the daughter cells. I've sort of made a mark on the chromosome where the change is. And as that gets copied, the chromosome gets copied, that change gets copied as well and exists in both of the daughter cells. So there should be no dilution of effect. However, as I mentioned before, with most gene editing technologies, there is this cutting enzyme required to make that cut in the DNA in order to change it. And there's a potential issue with that, in that if the cutting enzyme is not 100% precise in the exact location where we want it to go, it could cut areas that you don't want cut and potentially cause other problems. So what are we trying to do at LogicBio? So the hope with the technology that we're using with GeneRide is that we can get that long-lasting effect, the durability of gene editing, without having to use one of those cutting enzymes. And so I've included some semi-fancy scientific terms for these concepts, like integrating. This means that the DNA in the chromosome itself is changed. It's integrated into the cell's own chromosomes, and it's nuclease free, so it does not include that enzyme that makes the cut in the DNA. I mentioned we call this technology GeneRide. It's taking a ride along with another gene on the patient's chromosome. And the way it's designed to work is shown here in the cartoon. This is the AAV that delivers the working copy of the gene to the cell. The working gene here in red or orange gets matched up with the patient's DNA. And here it takes advantage of a natural process. All of our cells have it. It's used for DNA repair. It's called homologous recombination. And what it does is, as it lines up with the patient's DNA, it gets incorporated without having to use a nuclease to make a cut. And so if this works, we think it addresses these two issues that I've mentioned, the durability and the safety due to nuclease effects. And we think this is especially important for kids because they're growing and the need for a durable treatment is really important, and of course a safe treatment. But of course, we need to do those trials to test if it does work. We've already been studying GeneRide for MMA for several years in animals. This is the preclinical phase of research. This slide is not specific to LogicBio or GeneRide. This is just how clinical development is done. There's several years of animal studies to generate some safety data, to generate efficacy data on the animal, and that will tell us and regulatory agencies like the FDA that it is safe enough, likely to be safe enough, and likely to be efficacious enough to test in humans. In more common diseases and drug development, there's a phase 1 clinical trial to test for safety in healthy volunteers, in other words, people who don't have the condition. And then once it's safe, shown to be safe in that group, it's moved on to phase 2, which tests for safety in people with the disease. In rare diseases, and particularly in gene therapy, these two phases are combined. It's not tested in patients, people without the condition. And this is the trial that we'll talk to you about today. It's a combined phase 1/2. If the phase 1/2 shows efficacy and safety in this small number of people, then it advances to a larger study where you're really testing for efficacy that will then determine if the drug gets approved or not. And occasionally after approval, further studies are needed to answer any questions that still exist. And so for our gene therapy for MMA, it's called HLB01 right now. We're really excited. We'll soon be starting a combined phase 1/2 trial. It's called Sunrise. And as I described, phase 1/2 are mostly looking at how safe the drug is, but also for signs of efficacy, like a change in certain chemicals in the blood. In MMA, methylmalonic acid will be measured to see if we can have an impact on that. Because HLB delivers a working copy of the mutase gene, I didn't go into details in that pathway, but there's several genes that can be involved in methylmalonic acidemia. HLB001 only contains the mutase gene, so it's not indicated for other causes of MMA. That will require a different gene in the gene therapy. We'll be enrolling patients aged 6 months to 12 years, starting in those 3 to 12. And like any trial, there are several other criteria that will need to be met, including if you've had a liver transplant, you're not eligible. These are where the current sites are for the clinical trial. These 7 centers of excellence across the country, where there are MMA experts at each of these sites. We tried to get a wide geographic distribution in the country, but for families that don't live close to one of these sites, travel and associated expenses can be covered. I think we probably have families on the call outside of the US. We don't currently have sites outside the US yet, but we would consider international patients if there's willingness to travel to the US for therapy. So lastly, I just wanted to point you to a website that has a lot of helpful information on it, sunrisemma.com. It has information about the trial itself, but also I think a really cool and helpful video that explains gene therapy, a lot of the stuff that I was talking about today. I think it does a really good job of explaining gene therapy. And that was all the slides I had. I think I'll pass back to Jamie to lead a Q&A session.
Great. Well, thanks, Daniel. And there were a number of comments along the way about the clarity of your presentation. And I know it's complicated information, so great job. We do have a number of questions that have come in during the session. And there's a few other questions that we have that have just come in recent days since we launched the Sunrise clinical trial website. So the first one is, Daniel, can you just explain why individuals who have had a liver or kidney transplant are excluded from the trial?
Sure. So it's a really good question. So if you think about what gene therapy is trying to do, which is to deliver a working copy of the gene to the liver cells, it's basically, in a way, the same thing that a liver transplant is doing, just in a different way. You're actually transplanting the cells themselves into the body. So once a person has a new liver, they already have the gene to produce the mutase enzyme. So the rationale for a gene therapy treatment is not really there.
Okay. And a couple of questions are coming in about inclusion, additional inclusion criteria. Can you explain why the cutoff would be 12 years of age? Like, why would we stop at that age group?
Yeah, we get that question a lot because I know there are a lot of teenage and older patients and families who are eager for a new treatment. So when LogicBio was getting started in MMA, they had a lot of conversations with physicians and experts in the condition and were advised that to make the most difference, those patients that are most likely to benefit from a therapy like this, you should intervene, you should treat as early as possible. This is why it's on the newborn screen. And one of the reasons we're so excited about the potential for GeneRide, because we are able to treat the very young. And so when we were designing the preclinical experiments, the treatments in the animals, we used very young animals, just born. And so the data was generated from the very young. And so the clinical trials, once you go to humans, is based upon the work that's already been done in animals. And so we're starting in a young age group. And that was, like I said, intentional because we were that's how the program was designed. Now, having said all that, the development program does not end with or even the first set of trials. And so if the treatment can be shown to work in this age group, there's the potential for going older and also for younger, for 0 to 6 months. And so the story remains to be told. We're just starting in this age group.
Okay. And I mean, sort of more generally, are you able to just run through some of the inclusion criteria for folks?
Sure. Maybe some of the more important ones. In any trial, there's a bunch of these, but we've already mentioned the transplant as an exclusion criteria. You cannot have had a transplant. We also want to be sure that patients are not too mild, that they have some features of the disease. This is something the FDA was very eager to make sure we included, that a certain level of MMA, certain clinical manifestations that will not be surprising to anybody on this call, but it needs to be a patient who would need this kind of therapy in the real world. We also, there's some lab tests to make sure that the liver is functioning okay, that the pancreas is functioning okay, just to make sure, or to try to minimize the chance of a safety issue happening.
Okay.
I don't know. Sorry.
No, good. I was going to just let people know. So if you're interested in seeing the full inclusion and exclusion criteria, you can visit the clinicaltrials.gov website, and full criteria is listed there. And we'll put that website up in just a few minutes. So there's a number of additional questions about the trial, and then there were a few from earlier on that I want to make sure we get to also. But can you give a sense of timing in terms of when this phase 1/2 trial would potentially be completed and we might know results from the trial?
Sure. So we're currently in the stage of the clinical trial where we're getting the sites up and running. So there's a process that has to be gone through to allow sites to start to enroll patients. And we expect certain sites to be able to enroll patients into the trial within a couple months. And so when we will get results depends upon how quickly we can enroll the trial. Obviously, each patient in the trial will be followed for 1 year. But if it takes a year to enroll the trial, then it's a year from when the last patient has been enrolled. And then there'll be some time to analyze the data. So I expect that along the way, we'll be able to make some kind of interim announcements about the data, but the real final analysis of the full data set will be a couple of years or so away.
Okay. And can you just clarify again, I think you mentioned it, but a few questions have come in, just what segment of the MMA population the trial is intended for?
I assume you mean genetically?
Yeah.
So because HLB001 includes the mutase gene, it's mutase deficient MMA patients. So you guys maybe are probably familiar with the terms MUT-, MUT0. These are patients who either have no mutase activity or a reduction in mutase activity. It's those patients, not at this time anyway, patients who have mutations in the genes involved in vitamin B12 metabolism or other causes of MMA.
Okay. And in terms of, and I know it's early because we're just getting started with the phase 1/2, but the potential that if the therapy proved to be safe and efficacious, could there be an extension of potential use of this to people who have had a transplant in the future and or to other segments of the MMA population?
Yeah, I mean, I don't want to make too many promises here, but sure. I mean, if it's demonstrated to be effective, then we'll be thinking very seriously about how to whether to expand it into other groups of people. I mean, our goal, of course, is to try to treat as many people as we can, no matter where they are or how old they are, et cetera. So if the question is there a chance? Yes, for sure.
I'm going to pivot back to just some of the earlier slides and the overview of gene therapy and gene editing. Is there an optimal age for someone to receive a potential gene therapy?
Do you mean for our gene therapy or gene therapy in general? Because the answer may be different.
Different. Why don't we answer, if that individual wants to write back in and clarify, but why don't we answer for this gene therapy?
I was actually going to start with
Oh, the other
any gene therapy. only because it was a major point that I was making in the presentation, which is the idea about dilution. And this is why you see a lot of the gene replacement trials in adults. So even in some metabolic conditions where children are affected, these trials take place in adults. And one reason is because there's less of a chance of that dilution because the cells aren't replicate as much. For LB001 and GeneRide, we think it's appropriate for kids. I mean, this is one of the potential benefits of this technology is it could be durable, and so could be used in children. And in MMA, as I mentioned on one of the earlier questions, we think and have been told by our experts that the earlier the better. So if we can help prevent some of the metabolic decompensations that cause a lot of the problems in MMA, then that can only be a good thing, and we want to intervene as early as possible.
Great, thank you. And then another question that's come in. So would HLB001 only affect liver cells or all cells in the body, given that MMA is throughout the body?
Yeah, that's an awesome question, and pretty advanced. So for any gene therapy, the cells that end up getting corrected depend upon where that AAV virus or whatever virus is being used, where that goes. And so there are different types of AAV. I didn't really talk about that, but there's different minor changes in the AAV that determine where in the body it goes, it's targeted to. And the particular AAV that we're using is targeted almost exclusively to the liver. And so it'll be those cells in the liver that get corrected, not elsewhere in the body.
Okay. And in terms of possible expansion of the trial, if the phase 1/2 proves to be successful and we advance to a phase 3, do you anticipate that might have trial sites outside of the United States?
Yes. Almost certainly we'll have trial sites outside the US.
Okay. And then one clarifying question about the potential for gene therapy. So will it stop the production of the buildup, or will it potentially stop the production of the buildup of methylmalonic acid, or will it prevent the production of ammonia?
So I don't totally understand the question, but if you think back to that slide with the pathway, what we're doing is trying to do is to replace that mutase function. So the block won't be there anymore. And so the methylmalonyl CoA won't accumulate, won't get converted to methylmalonic acid. So I think I'm thinking back to the wording of the question, it would reduce the buildup of MMA by helping to restore that breakdown. I'm not sure I got that one, so
Okay.
maybe that person can contact us separately.
Okay. And I know you've addressed this a little bit already, but the potential for GeneRide technology to be applied to other organic acid disorders such as propionic acidemia.
Yeah, so one of the good things, great things about GeneRide is that it has the potential to be used in other diseases. The only thing that needs to happen is that you put in a different gene. And so potentially diseases like propionic acidemia, you change the mutase gene out, put in a different gene in, and you have the potential to address other diseases. And we're doing work in the laboratory now on other diseases. So for sure, it's adaptable.
Okay, and it looks like we have just 2 more questions. Okay. How soon into this phase 1/2 trial might we take the younger cohort of 6 months of age?
Oh, yeah, that's another good question. I didn't go to the details of the design of the trial, but the first 2 patients in the trial need to be in that 3 to 12 years of age. And once we have some data about safety and that we have some evidence that the gene therapy is doing what it's supposed to be doing, then the 3rd patient in the trial could be as young as 6 months of age.
And the last question that we have is, again, related to the trial. Would we accept patients who have received mRNA after a washout period?
Who have received mRNA? I think it's one of the, Marie, I might look to you, but I think it's one of the exclusion criteria that if you've received other genetic therapy for MMA, you're not eligible.
That's correct.
Okay, and then just, sorry, one last quick question. When we say the cutoff age is 12, is it under the age of 12 or does it include the age of 12?
It includes, it's 12 years and 364 days.
Great. Well, those were fantastic questions. Sorry, go ahead.
No, I just want to make sure I didn't say anything wrong. That's right, Marie, isn't it?
That's correct.
And I could just ask one question, that this is a one-time treatment, correct?
That's correct. That's correct. A single treatment. Patients would come into the hospital to receive the treatment and then would follow up with their physicians throughout the trial. But it's given one time.
And it's given one time because that's all it needs to do its thing, right?
Yeah, if it does what it's supposed to do, you give it and it's there, and you don't need to give it again.
Well, those were fantastic questions, and Daniel, thank you. That was sort of rapid fire, and you did a great job answering those all. Do you mind going to the next slide?
Oh, yeah, sure.
Great. So we just want to make sure as we wrap up, because we're just about at 12. We want to make sure you have resources to help answer additional questions and to learn more. So of course, if you have potential questions about participation in the trial, we always encourage you to speak with your healthcare provider or your child's healthcare provider. As we referenced, the website clinicaltrials.gov is a bit of a clearinghouse for clinical trial details. So this trial is up on that website, and that's where you can find out more details about the different sites. We had the list up, and that list is also on the Sunrise trial website. But it has the full list of inclusion and exclusion criteria, and again, more details about the goals of the study. So that's another place to go. As you see here, this is our Sunrise clinical trial website. And so that was just launched about 2 weeks ago. And I know some of you have probably contacted us through that site, which is great. And you are able to send us questions through that site. And lastly, we have our corporate website. So for those who are just looking to learn more about LogicBio and our work, logicbio.com is the website. And just yesterday, in advance of this webinar, we actually updated our patients and families page. So that is where you can sign up for updates from us. You can send us emails. So if you have additional questions stemming from the conversation today or just in general, you can go to that website. We also have the Understanding Gene Therapy video posted there and some family stories, including a story about the Stagny family. So please feel free to check that out. And again, you can sign up for updates. And if we go to the last slide, I just wanted to give you our email address. So again, if you have specific questions stemming from today or are looking to get in touch with us, this is the best way to do that. I check that email throughout the day and we try to get back to folks as quickly as we can. As I mentioned at the outset, we are really keen to engage with the MMA patient community. So we really do welcome hearing from you, whether you have a question, just want to have a chat with us and tell us about your family and your experience living with MMA. Please feel free to reach out. Don't be shy. It really sort of our daily purpose in our work is for you and about you. And so we really want you to be a part of that process and encourage you to engage with us. And I, again, just want to thank Dr. Gruskin for your time and the presentation. And really just want to thank Kathy and the OAA team for offering this opportunity. This was really helpful for us and exciting for us. And we appreciate you setting this up and helping the community to participate. And thank you for your ongoing partnership as we work and endeavor to hopefully bring a therapy to the community. Thanks everyone for joining, and I'll turn it back over to Kathy.
Well, thanks so much for speaking with our families today. And I have, like I said, recorded this session. I will share it on Facebook and pass that link out from YouTube, put it on YouTube so that you can share it with families that weren't able to attend or just go back and review it. Like I said, we value our partnership with our pharma companies, LogicBio being one of the first ones to reach out to us. So if you have questions, feel free to contact them. They're the experts. I am probably not the best person to ask some of the details. However, I'm happy to share my what I know as well if you want to contact me. So again, thanks so much to LogicBio, Dr. Gruskin and Jamie and Marie for spending some time with us on Saturday morning. Appreciate taking that time out, and hope everybody has a great day, great weekend, and stay safe. And we'll talk to you soon.
Great. Thank you.
Thank you.